I'm at Virginia Tech, and I'm excited to talk to you about poison ivy. Hopefully I can convert you that it's a great system, a wonderful plant. It's very beautiful, right? So some of the things that, I'm going to try to stay in front of the microphone, but I'm kind of a wanderer. Some of the things that are great about poison ivy are that it's really long-lived, right? So 35 years, develops these really large lianas, and if you wear the proper personal protective equipment, you can actually do some woodworking with it. No? Okay. So, some of the facts that folks actually don't know about poison ivy is that it's actually dioecious, right? So here's some male flowers. So you have separate male and female plants. It produces these drupes, which are typically bird dispersed. All sorts of birds love these drupes. It's also really subject to herbivory. So that's sort of counter to everyone thinks that these compounds that it makes us itchy are actually defensive, and the jury's still out. We don't know about that, but certainly no other mammals are affected by it other than us. And of course that's why most of us know it, right? For this contact dermatitis. It actually increases in sensitivity as you age, you get more exposure to it, you increase your sensitivity. So those of you who think, oh, I don't get a reaction, you will one day if you keep rubbing it on your arm. So the itchiness or this contact dermatitis is caused by not one compound, but a whole class of compounds that we collectively call urushiol, right? And it's the basic ring with the alkyl side chain. One of the things that I got excited about, those of you who know me well know that this is the part that I really care about, but it is these compounds, right? Can we actually find natural variation in the wild and figure out what these compounds are doing? And it turns out, no. They are very, very environmentally plastic, right? The compounds, the amount of substitution changes, those things all change. One thing that's actually consistent is male versus female plants. Right? So they use a different monomer, whether they're male plants or female plants. So that's something that we can move forward with. Here's the most recently published phylogeny of the Anacardiaceae. What I'm going to highlight is that toxicodendron forms this nice little monophyletic group, although the support for separation within toxicodendron is varying. And that's in part because the last treatment of it was by Gillis in 1971, who characterized most of the relationships morphologically, but settled on these three species for North America, which is the western poison ivy, toxicodendron rydbergii, poison oak, diversilobium, and of course radicans, eastern poison ivy. The radicans is the one I'm going to spend the rest of the time talking about today. So what we wanted to do was go out and look at the distribution of poison ivy and see if we can actually develop some genetic markers and do some population genomic studies within them. That meant we needed a genome. Number one, because there was no published genome for anything in the clade, right? So cashew and mango are also in this clade. I've been told by the group that's sequencing mango that they're going to be releasing it. They've told me that every year for the last 3 years. So hopefully one day they will. We'd like to know something about population structure and history. We'd really like to develop gene models for some of the informed ecological studies we'd like to do. I'm not going to tell you the secret that I'm not going to tell you, which is that this plant takes 2 to 3 years to mature to be able to make crosses and make segregated populations. So it would be super nice if we can do some GWAS, although John Slade says that's going to be kind of difficult in natural populations. Hopefully we'll get there. Finally, identify genes associated with urushiol production. So urushiol is this lacquer compound. Urushiol is named for the Japanese lacquer tree, which is of course in the toxicodendron family. It's a nice hard coating. What's really useful for are people who make tires, apparently. It is a hardening agent that can add life to the rubber and therefore add tread wear. And so they'd really like to understand what the genes are involved in urushiol production so that they could sort of make better tires. Didn't know anything about that until I started this project. Today I'm just going to focus on the first two, which number one, that there's no published genome in the clade. We're going to see if we can resolve that. We're going to try to do it for just $5,000. because that was my budget. And so the first thing that I want to know as a genomicist is how big is it, right? And so I didn't know going into it how big it would be, so I used Arabidopsis and I used tomato as standards to get an estimate of the genome size. The problem was that I asked for Arabidopsis from a person who studies developmental biology. And so I was kind of terrified when the blue peaks there are for Arabidopsis. That should be one peak because Arabidopsis is diploid, right? Well, no, it actually undergoes in some of the colline leaves, it undergoes endo-reduplication, and therefore we have multiple ploidy and that Arabidopsis problem resolved. Not a technical issue, but one to think about. So here we are with radicans, somewhere around 462 megabases. So we'll call it 500 just to make it even. And we generated a lot of PacBio data from 15 cells. And then because I had a nanopore, I tossed in some nanopore reads that were about, trying to get some really long reads, 100 kb reads or such, because the PacBio was a little bit small. We assembled it using multiple different platforms. The one I'm going to describe is Canoe, which gave us the most consistent assembly. Once we, so here are the raw contigs, the primary scaffolds with just PacBio, and then adding some of those nanopore long reads. And what we end up with is about 3,700 contigs, a total length of 404 megabases, the longest contig 1.54 megs, and 50 of 204 kb. So these are sort of middle of the road statistics, right? They're reasonably good. We can move forward. The thing I'll point out for you is, I said that our estimate was 462 megabases. We're at 404, so probably there's some repeats in there that we're not capturing. Our PacBio reads are not quite long enough, so we're collapsing those. I'm okay with that. We wanted to sort of get an overall assessment of the integrity of the genome. So we used the common tool BUSCO to characterize that. The important number here is at the top, about 84.7% BUSCO completeness score. To give you some idea, we'd like it to be 90 or higher. The tomato genome, when I just run it through BUSCO, it's a relatively complete genome. You get about 96% BUSCO completeness. So we're missing some genes. We're not capturing the full gene space. Still, we can annotate it. Because of another project, we had access to a lot of RNA-seq data from this same species, the tiger people. They were trying to find genes without any sort of framework to put it in. We used Augustus to train with Arabidopsis, standard things. And then we used a program written by a friend of mine, Simo Zhang, called Agouti, to actually go in and chop up our assembly and try to realign to get our annotation models, our gene models, really correct for their start and stop sites. And so that's what Agouti does. Agouti then also helps us with those reads that were collapsed if we could try to separate them somewhat based on start-stop. So when we do that, the big thing, we spend, almost 2 weeks trying to annotate this thing to get this number right here. Right, so about 22,843 genes, to just give you some idea of a sanity check. Here we're sort of around the norm. So these are the closest relatives that I could find with published annotation data. So it seems fairly reasonable. Certain we're missing some genes. Okay, so we have a genome now. It's a draft genome. It's not complete, but we're working on completing it. We're going to do some bionanos. So we can try to get this thing into pseudomolecules and some just straight up regular DNA-seq so that we can improve our gene models. But let's do something fun with it, right? Something interesting, because what I really care about is the ecology, right? And one of the reasons to do that in this species is it's sort of been a bellwether of climate change in the past. So research from a few years ago showed that the toxicity of poison ivy is increasing. So that one of the particular alkyl chains that gets substituted on is actually increasing in frequency under experimentally elevated CO2, right? As is growth rate. But I'll remind you, I'm actually interested in herbivory. So I'm going to get back to that part. So can we actually go out and start to do population genomics in this species? Well, I wanted to do that. Got a team of faculty together, and we went out and sampled 80 locations, sampling every 1 to 2 kilometers. We included some divergent samples, so from states away. Collected all location information. We used GBS to generate about 7,000 markers. So it's a reasonable marker set to go out and look. Now, if you're familiar with North America, you might be familiar with Blacksburg. If you're in Blacksburg and you want to go sampling poison ivy, best place to do that is the Appalachian Trail. Right? So we can go out and hike along the Appalachian Trail. One of the reasons that I wanted to do it is the Appalachian Trail actually pairs with well, you can't see it in this version. There's a yellow line there that is the Blue Ridge Parkway, right? And so it's a nice paired sample where I have elevated CO2 from all the cars driving down the Blue Ridge Parkway and the Appalachian Trail. The other thing that's interesting is when you ask the National Park Service for permission to collect, for a permit to collect poison ivy, they say, you don't need a permit, take as much as you want. So we go out and we collect, we collected across here. The reason that we limited it to 80 is because no one else wanted to carry drying silica 15 pounds worth in their backpack. I don't quite know why, but that's what we had to do. So we did that just to give you a visual on sort of the topography, what it looked like. This is some of our collection points. You can see it moves through wooded and non-wooded developed areas. Right? The Appalachian Trail is not this pristine walk through the woods the entire time. One of the problems we have here, so there's lots of variation in growth habit, climbing, vining, these small shrubby plants, but then these giant collections. And the real question here is, looking at this, is this one plant or is this many plants? Well, we can use our GBS data to answer that question. You don't really have to be an expert to see here that these ones with all the same numbers where I collected all along that vine, they tend to be the same thing, right? So we have to correct these data in our population structure analyses. We have to correct for clonality. And when we do that, we actually find 3 distinct genetic clusters within our sampling locations. That agrees with a structure analysis, and when we try to plot our bar plots, by geographic order, so this is latitude, doesn't really make any sense. When we add in an eyeballed habitat, right, so we're collecting habitat information, we see that there is some association between everything down here that starts with a D indicates disturbed, right? So it was disturbed and open, disturbed and developed, or disturbed and forested. And so we're seeing some sort of association there. Unfortunately, my p-value is not quite 10 to the minus 253, so we're going to keep on collecting until we can feel very confident. Right? So our goal is to get this work funded and move out and collect hundreds of samples so that we can really get a better picture of what's going on with this plant that everyone knows and sees, but maybe isn't so familiar with. With that, I'd like to acknowledge my collaborators, Ruliano and John Gillesco, and then folks that hike the AT with us. This is John does some of the Urushiol work. This is a caricature that someone did of the poison ivy man. So,